right, so now you can tell all your friends and family that you know how to draw the structure of DNA if you want to. If you just copy whatever I have up on the board, you could just keep extending it and making it longer and longer, and boom, you've got the structure of a strand of DNA. How do we know this is DNA? Take a look and see what I have up on the board. Can you recognize that you have these two sugars here, and these two sugars do not have an OH on carbon number 2? Can you see that they must be deoxyribose? What's another way that you know that it's a strand of DNA? Can you recognize this cyclical amine base over here? Can you see that it's a pyrimidine with 2 carbonyls, but it has that extra carbon attached? So this must be a thiamine, a T, okay? Does anyone recognize what this is? This is also a pyrimidine. Remember, I taught you there were 3 pyrimidines. There was thiamine, uracil, and cytosine. Remember that? And we said that both thiamine and uracil have the carbonyl groups. Well, obviously it can't be uracil, right? Because uracil, you wouldn't find it in DNA. This must be cytosine. So yep, it is cytosine. So this over here is DTMP, which is deoxythymidine monophosphate, and this is DCMP, deoxycytidine monophosphate. Now this is what I wanted to show you guys. How do we connect these things? Remember how I told you it's a sugar phosphate backbone? Here's a phosphate, here's a sugar. Here's a phosphate, here's a sugar. If I wanted to make it longer, I'd put another phosphate and then a sugar, and then another phosphate and then a sugar. So it'll always be phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, etc., etc. And then coming off of the sugar is the cyclical amine base. Now, this is considered a dinucleotide. Why? Because there are 2 nucleotides. The hypothetical one that I did at the end of the last video, that was a trinucleotide. I did this little box figure thing. That was a trinucleotide because I pretended there were 3 nucleotides here. But here are actually 2 nucleotides. So this first one is 1 nucleotide. This is a second nucleotide. Let's see where they got hooked up. You see carbon 3 of this pentose. So this is carbon 1, this is carbon 2, this is carbon 3, this is 4, and this is 5. So at carbon 3 of the deoxyribose, you're going to remove an HOH. So you remove an H from here, OH from here, always removing a molecule of water. And then the phosphate of this nucleotide hooks up to carbon 3. Okay, so that's what DNA strand looks like. That's what an RNA strand will look like. I can draw another one down over here. If it's an RNA strand, let's say you do a dinucleotide for RNA, you would notice that there's an OH here at carbon 2. But in both DNA and RNA, the phosphate group is hooked up to carbon 3. Okay? Now, for those of you who've had a biology class, have you heard of the term 5' 3' or 3' 5'? Does that sound familiar? If you've had an A&P class, you probably had that, or microbiology. What do they mean by that? Well, this end of the molecule where this carbon 5 is not hooked up to another nucleotide, this is called the 5' end. And they call it 5', not just 5, because this cyclical amine base is numbered 1 through whatever, 9. And then this one is 1 through 6. This is 1', 2', 3', 4', 5'. So this is considered the 5' end, the end where this phosphate group is not hooked up to a sugar on top of it. And then down over here, where this sugar doesn't have another phosphate group coming underneath it, this is called the 3' end. Okay? So when we're talking about replication, transcription, translation, all that stuff, we start using the terms 5', 3' to orient the strand. Okay, remember at the end of the protein chapters, I was telling you sometimes they call this end the N terminal and this end the C terminal. That's just to orient the strand, the polypeptide strand. Well, in DNA and RNA, they always orient the strands by saying this is the 5' end and this is the 3' end. Okay, so again, you're not going to have to draw this, but you'll have to recognize it. So if I have a structure for you guys and it's a dinucleotide, you'd have to tell me that it's a dinucleotide. You'd have to tell me it's a dinucleotide found in DNA or RNA, and you'd have to name the actual nucleotides. You'd have to tell me, oh, this one over here is DTMP. You can use acronyms if you don't want to use the full name, or you could write down D-thymidine monophosphate, and this one would be D-cytidine monophosphate, or DCMP. Okay? All right, so on the list of things, actually, so we just drew a strand of DNA. If I wanted to turn this into a strand of RNA, this is what I would do. I'm going to remove this. So now what do I have? I have a UMP. I'm going to throw an OH here and put an OH here. Remove this D. And I just switched a strand of DNA, and now it looks like a strand of RNA. So it's the same sugar phosphate backbone, but now instead of a deoxyribose, I have a ribose. So this now is a dinucleotide found in RNA. The one I had drawn earlier, dinucleotide found in DNA. How did you know? The sugar, okay? And then here you have UMP. Now, if I didn't have uracil or thymine, if I just had, let's say, a guanine and an adenine and a cytosine, those won't tell you if it's DNA or RNA. What will tell you is the sugar. So if I have those guys hooked up to a ribose, you know you've got an RNA. If I have those guys hooked up to a deoxyribose, you know you have a DNA. Okay? All right. You guys have had, or you've heard of genetics. You know that in order to have the cell cycle in a biology class, you know that before a cell divides, it has to make a copy of all of its DNA. So like the mother strand becomes, and you have the mother cell with the original DNA, and then you have two daughter cells with identical DNA. So in order for any type of replication to occur, meaning making more cells, you have to make copies of the DNA. So we're going to do a hypothetical. So we're going to say that we have a double-stranded DNA. Let's pretend that this is a double-stranded DNA. So we have A, T, C, G, A, C, C, T. Let's pretend this is just a hypothetical strand of DNA. Remember how I told you that DNA, unlike RNA, is double-stranded? So there's something called complementary base pairs. Complementary base pairs. Anytime you have an adenine, you see a thymine on the other strand. And between adenine and thymine, you have two hydrogen bonds. And then anytime you have a guanine, you have three hydrogen bonds to a cytosine. Three hydrogen bonds. Okay? So, if I have a second strand here, this would be a T, because opposite the A has to be a T. This would be an A. This would be a G. This would be a C. This would be a T. This would be a G. This would be a G. And this would be an A. Can everybody see what I'm doing here? So this is a hypothetical double-stranded DNA. I could draw the whole sugar phosphate backbone, but we're just focusing on the cyclical amine bases being attracted to each other. So if I had this as one strand, this would be the other strand. So let's say I have an A here, thymine here, G here, C here. So we've heard of the double helix. This is your DNA. So sugar phosphate backbone is all of this line, and then the adenines are pointing out and the thymines are pointing in towards each other, and then they hydrogen bond with each other. Okay? And anytime you have something like this, double-stranded, one side is 5', 3', the other side is 3', 5'. So they always complementary base pair, opposite. Okay, so one strand goes in one direction, the other strand is in the other direction, they face each other, and then there's hydrogen bonding between the cyclical amine bases. Okay, now the term replication means we're going to replicate, we're going to make more DNA. How does that happen? Just a quick, simple way of looking at it. You're going to open up the DNA, uncoil it. This DNA is coiled. We're going to uncoil it. There's an enzyme. You don't need to worry about the name of the enzyme, but it's an enzyme called helicase that opens up the two strands. So now I have, because remember, they're hydrogen bond. They're attracted to each other. So now I have A, T, C, G, A, C, C, T, 3'. And then opened up, over here on this side, I had T, A, G, C, T, G, G, A, 5'. Okay, we unzipped it, we opened it up, we broke the hydrogen bonds. Now you get these nucleotides that your body has just synthesized. Your protein in your body, you make the cyclical amine bases, the sugars are synthesized from glucose, the phosphate, you're eating phosphorus, so you're making these nucleotides, and now these nucleotides are going to come and line up here. Separate nucleotides, and the right ones are going to go in front of the right ones, and then over here also you're going to have an A, a T, a C, a G, an A, a C, and a T. And then you get an enzyme called DNA polymerase. It zips it all up, forms the sugar phosphate backbone, and then zips this one too. Sugar phosphate backbone. You already have all the hydrogen bonding done. Take a look at these two and compare them to the original. Identical, right? Now your cell can divide and you have two new cells. So this was the original cell. It replicated its DNA, and then you divide, and now you have two new daughter cells. So they say the mother cell breaks down into two new daughter cells that have the identical DNA as the mother cell. So that's replication. Okay, next video we will start transcription and translation.